Good evening, all. Welcome to the sixth live session on Fundamental of Protein Chemistry. I hope you all are enjoying this course, which is Fundamentals of Protein Chemistry. And now we are in week 6. I hope I am audible and I'm visible enough. Okay. So now we are in the week 6, which is on the enzymes and enzyme mechanism. And till now we have covered week 5 till week 5 and all the I hope you all have gone through the lectures material that have been uploaded in the NPTEL portal, and also you have submitted the assignments till week 5. Okay, the evaluations have been done till week 5, and now we will be discussing the assignment problems, sample assignment problems of week 6 uh, module. So let me share my screen and then we will start the meeting. I hope my slides are visible to all of you. And I again welcome you all in this live session. I am Karan Kumar, PMRF scholar at IIT Guwahati, and you must have covered the concept that covered in the week 6, where the enzymes, its importance, and the classification of the different enzymes. Then you must have covered the enzyme substrate interaction, then substrate and geometry specificity, which is very much important for any reaction to happen or any enzyme substrate interaction to happen. Then you studied about the factors influencing the enzymatic activity and how to regulate those factors, how to regulate the enzyme activity. Then you have studied about the energy diagrams, then mode of catalysis. There are various types of catalysis mode, such as acid-base catalysis, covalent catalysis, metal ion catalysis, electrostatic catalysis, so and so forth, and the various catalytic mechanisms you must have studied in the week 6. So the important keywords that were in the week 6 are the catalysis, then various terms related to the enzyme and its classification, such as oxidases, transferases, and various other 6 different classification of the enzyme. You must have studied that. Then the terminology related to the protein or enzyme, such as coenzyme, protein cofactors, then proaesthetic group, then holoenzyme, and difference between the holoenzyme and apoprotein. Then you must have studied the transition state, its chemistry, how enzyme helps to lower down the energy of transition state, and then finally make the reaction feasible in the forward direction. Then you must have studied the catalytic mode, then how the inhibitors inhibits the enzyme, uh, and the different fashions of enzyme inhibition. Then mode of interaction such as induced fit. Sorry, I have written here is the field which is induced fit. Let me correct it. Uh, okay, this is induced fit. Okay. Then you have studied about the lock and key mechanism. Lock and key mechanism. Then about the catalytic triad and binding pockets, active site. You must have come across these important keywords which were related to the week 6. Now coming to the contents of this PPT, what I'm going to present today. As always, like I will be presenting the some practice question on current module, which is on week 6. Then I will discuss some important topics and I will take questions from you if you have any doubts related to the content or the related to assignment that we just discuss. We will discuss. Then also about the previous week's questions if you have. Then we will discuss about the scope of this module with research perspective like as always we discuss about every week. Now coming to the questions of this week, the sample problems. Your first question is the active site of lysozyme contains a glutamic acid residue, which is having a pKa of 5.5. You must have remember. I hope you have remembered what are the pKa of amino acid and how it is important to know which charge it will possess in that particular pH or at the isoelectric point. Then this lysozyme contains a glutamic amino acid residue. Then also it contains an aspartic amino acid residue, which have the pKa value of 4. Okay. Now the question is given: the optimum pH is centered around 5. Okay, pH 5. Then your question is to find out the correct statement. Which statement is correct about the mechanism of lysozyme? Like, then your options are: the glutamic acid residue is a more polar environment than the aspartic acid. Then option B is: during the entire catalytic mechanism, the aspartic acid amino acid residue remains unprotonated. Then your option C is: during the mechanism, the oxyanion transition state forms. Option D is: the glutamic acid residue act as a general base catalyst. So I will give time to think about it. So think about this question and then try to come up with some good approach. So anyone willing to answer this question? Okay, pause for a second and then think about this. The question is regarding lysozyme, and the question has given it is talking about the two amino acid residue that it contains. Both of them are the acidic amino acid residue, right? A glutamic acid and aspartic acid, right? Then it is given that glutamic amino acid residue has the pK value of 5.5 and aspartic amino acid residue has a pK value of 4. And the optimum pH for this enzyme is around 5. Okay. Then your question is filter out the correct statement about the mechanism of lysozyme. And your options are: the glutamic acid residue is more polar environment that of the aspartic acid. Option B is: during the entire catalytic mechanism, the aspartic acid residue remains unprotonated. Option C is: during the mechanism, the oxyanion transition state forms. And option D is: the glutamic acid residue act as a general base catalyst. Any such any comments on this question? So here, let me discuss this question. Here, the glutamic amino acid residue given pK is 5.5 and the environmental pH is 5. So in that condition, the glutamic amino acid residue will not lose its proton. It will keep its proton and it will remain in the protonated state, right? However, aspartic amino acid residue has a pK value of 4. Okay. So at that pK, when the surrounding pH is 5 and the aspartic amino acid residue has a pK value of 4, then in that scenario, the aspartic amino acid residue will lose its proton and it will exist in the deprotonated state or unprotonated state. So your option B is correct, right? So during the entire catalytic mechanism, the pH is around 5 always. So in that catalytic mechanism, the aspartic amino acid residue will remains unprotonated state, right? Because the surrounding pH is higher than the pK value of aspartic amino acid residue. Coming to the next question, your next question is: salivary amylase is an enzyme in human that breaks down the starch. Okay, the statement is salivary amylase that found in mouth. Okay, that are found in the saliva. Okay, so is an enzyme in humans that break down the starch. The optimum pH, for where is the cursor? Okay, here. The optimum pH for the reaction is 6.7. The rate of this reaction would not be affected by. You have to find out the correct option for this, and your options are: option A, maintaining the pH of the reaction at 6. Option B, if you do not affect the substrate concentration or enzyme concentration or decreasing the temperature of reaction by 5 degrees Celsius. So what do you think about this question?
A.
A, okay. A is about the maintaining the pH of the reaction at 6.5, 6.7, right? Okay, why do you think so? And why other options are incorrect? Can you give an
So,
explanation regarding it?
because salivary amylase is an enzyme, and if we change the either the temperature or concentration, it will show a bifurcation in the reaction. But 6.7
Okay,
is its optimum pH, so it will not get affected.
okay, yeah, very good. So if we maintain the pH of the reaction at 6.7, because the optimum pH for this reaction to happen is 6.7, so this will not affect the rate of the reaction. However, in the other case, decreasing the temperature by any decreasing or increasing by any rate or any degree Celsius, like the temperature, that will surely affect the rate of the reaction, because rate depends on because uh, the rate is dependent on the temperature also. Similar to the substrate, this condition is also similar to the substrate concentration, where the initial rate, if you remember the Maclaurin constant, Maclaurin equation expression, in that rate depends on both the enzyme concentration and substrate concentration. That is why if we affect the substrate concentration or enzyme concentration, then that would affect the rate of the reaction. So the correct answer for this question is option A, maintaining the pH of the reaction at 6.7. Now your next question is tyrosine 2,3 amino mutase is an example of which kind of enzyme? Your first option is phosphodiesterase. Option B is intermolecular transferase. Option C is glycosyl transferase. Option D is methyl transferase. Any guess? Any guess about the tyrosine 2,3 amino mutase? One thing is clear, right? Like most of the options are here about the transferases, and only one option is diesterase. So, what do you think about the tyrosine 2,3 amino mutase? Any guess? Okay. This is a kind of factual question that you need to remember. The first thing is this, as the name suggests, the tyrosine 2,3 amino mutase means because this we can guess that it must be in the kind of what you say the synthesis, some kind of synthesis or synthesis where the tyrosine will be involved, right? So actually the tyrosine 2,3 amino mutase is a member of family of isomerase, and specifically they form the intermolecular transferase. They come in the category of the intermolecular transferase. And what does this tyrosine 2,3 amino mutase does? It participates in the tyrosine metabolism. Tyrosine metabolism, as the term as the term says, tyrosine metabolism. Metabolism is nothing but the formation and degradation, right? So that means this tyrosine 2,3 amino mutase is an enzyme where it will be involving with the tyrosine metabolism, and actually this is involved in the formation of the S beta, the compound which is S beta tyrosine. Okay, S beta tyrosine. Okay, and another fact about this enzyme is this is a intermolecular transferase, and like intermolecular transferase, and this transfers the amino group. Okay, this transfers the amino group. Okay, and also this is also called as tyrosine alpha beta mutase. Okay, so another fact is this is also called as tyrosine alpha beta mutase. Alpha comma beta mutase. Okay, so your correct answer here is it is an intermolecular transferase. Now coming to the next question. Choose the incorrect statement among these four. So your options are metal ions participate in the catalytic process by binding to the substrate to orient them properly for reaction. Option B, metal ions participate in the catalytic process by mediating oxidation reduction reaction through reversible changes in the metal ion oxidation state. Option C is metal ions participate in the catalytic process by tightly bound or stably associated with the enzyme. And option D, metal ions participate in the catalytic process by electrostatically stabilizing or shielding the negatively charged. So what do you think about this question? Any guess? Question number 4.
The B.
B option. B option is about first of all we need to identify the incorrect statement. Okay. So your option B is metal ion participate in the catalytic process by mediating the oxidation reduction reaction through reversible changes in the metal ions oxidation state. So are you sure about this option? Why other
Okay.
yes, yes.
I've
Why
guessed
this is
it.
incorrect?
So because mainly the means the side chains of the amino acid, they like histidine have the imidazole. These groups basically take part in the oxidation reduction reaction.
Mhm. Okay. So you think
That's
this
great.
option is incorrect. Okay. Any other answer you have? If there are other participants, they would like to participate in this question. Any other option I can get? Okay. Yeah. First of all, the about the metalloproteins in the when the metal is participating as a cofactor in that catalytic process, they might they might actually happen with the they can form the oxidation reaction. Why? Because in case of when the when the metal ions such as Fe3 plus Fe, iron, and other magnesium. Magnesium and zinc, these are mostly they hold the protein, but the ferrous ion, when it is present, they actually participate in the oxidation reaction through the reversible changing of its oxidation state because they have the different bands and they have different oxidation states, such as when the Cu2 plus also is present in the as a cofactor, that metal ions also participate in the formation of they also participate in the oxidation reaction. So option B is correct actually. But if you see, if you examine all other options such as metal ion participate in the catalytic process by binding to a substrate, this is very accurate option. So, but this is not inaccurate. So option B is also correct. Option A is also correct. Now talking about the option C, metal ion participate in the catalytic process by tightly bound or the stably associated with the enzyme. Uh, that might not be the case because mostly these if they are tightly bound or stably associated, then they would not be able to participate in some catalytic processes. They will be kept in core. However, what we see, the ions are present in the free part of the protein and they mostly participate by the formation of electrostatic interactions, By that fashion, they only do the they process through the catalytic. Perform the catalytic processes. They do not bound it tightly. So this option is incorrect. However, if we're talking about the option D, the metal ion participate in the catalytic process by electrostatically stabilizing or shielding the negative charge. Yes, this is very accurate because they participate electrostatically, not by tightly bound. So option C is incorrect, and the correct answer is C. Uh, uh, did you understand this, or you need explanation? Am I clear enough?
Yes, sir.
Okay. So we will move to the next question. The next question is, which of the following statement is true about the reference to enzyme? Okay. So is apoenzyme is equal to the holoenzyme plus coenzyme, or holoenzyme is apoenzyme plus coenzyme? Coenzyme is apoenzyme plus holoenzyme, or holoenzyme is equal to apoenzyme minus coenzyme. Which is the correct statement? For this, you need to have the clear understanding of
B.
what are the apoenzyme. Yes, option B.
B. Yes.
Okay, option B. Yeah, you are correct. Mostly, the whole part of the enzyme, like the complete enzyme with its cofactor and coenzyme, those are called at the holoenzyme. However, if a enzyme is lacking its coenzyme or cofactor, then it is termed as the apoenzyme. Okay, so option B is correct. So holoenzyme is apoenzyme plus coenzyme. Now your next question is, trypsin and chymotrypsin cleaves at the amide bond after the option A, C-terminal side of the amino acid and C-terminal side of the hydrophobic amino acid respectively. This option is talking about the like the first thing is about trypsin and second statement is about the chymotrypsin. Okay, your options are option A, C-terminal side of the acidic amino acid residue and C-terminal side of the hydrophobic amino acid residue respectively. And option B is N-terminal side of the basic amino acid residue or the C-terminal side of the hydrophobic amino acid residue. Option C is C-terminal side of the amino acid residue, basic amino acid residue, or C-terminal side of the hydrophobic amino acid residue. Option D is C-terminal side of the acidic amino acid residue and C-terminal side of the hydrophilic amino acid residue. So, according to you, what is the correct statement for this? Like, what is the cleavage site of trypsin and chymotrypsin? This is very much important. Is it acidic, basic, basic, acidic, or hydrophobic? For chymotrypsin, hydrophobic, hydrophobic, hydrophilic. Yes. Uh, any guess? Do you remember this? This I discussed in the week 0 assignment also about the trypsin. Like if you have gone through this, the same kind of similar kind of question you must have got into the week 0 assignment, where the you had been given a peptide sequence, and among that you need to identify the which is the cleavage site of the trypsin or how many fragments the trypsin digestion will do if it cleaves the peptide bond. Okay, any guess? What are the C terminal and N terminal? Can somebody tell what is the C terminal?
The C terminal is the end which is having COH2 and the N terminal is having NH3 plus 2.
NH2, okay. Amino group is N terminal and carboxylic group is the C terminal. Okay. So which is the left hand side and which is right hand side? What is the like nomenclature of writing a peptide bond? C terminal is on the left side or right side?
I'm not sure. It's, I think, right. C terminal is on the right side.
C terminal is, you are saying C terminal is in the right side. Okay. Any other guess? Any other anyone else would like to contribute? When the bond is being formed, then N terminal is in the left hand side and C terminal is in the right hand side. You are very much accurate. So here the correct answer for this question is trypsin. You need to remember these all because this is uh, like with respect to the all other competitive exam, these kind of questions are usually appear in every kind of examinations. Like any any kind of competitive examination, you will find this kind of question. So please try to remember the cleavage site of all proteases: trypsin, chymotrypsin, or pepsin. There are many more, uh, but the trypsin is very common one. Trypsin and chymotrypsin are very common one. So you try to remember this one because this will appear in all kind of competitive examinations. So trypsin have the like the cleavage site for trypsin is the C terminal site of the basic amino acid residue. Okay, and about the chymotrypsin, they usually cleave at the C terminal of both cleave at the carboxylic terminal only. Okay, but the chymotrypsin cleaves at the hydrophobic amino acid residue. So in case of basic amino acid residue, you will have that you will try in the next question. There is one more question that is there. This was in the week 0 assignment. From there, I have taken it. So now, based on the statement, based on the answer that I have given above, that C terminal side of the basic amino acid residue and C terminal side of the hydrophobic amino acid residue for the chymotrypsin. Now, can you answer this question? The trypsin digestion of MVDYD GK peptide sequence gives how many fragments? Can you attempt this question? You know that the trypsin will cleave at the basic amino acid residue and C terminal of it. So, what will be the correct answer for this? How many peptide sequence you will get? So among these peptide, like in this peptide, which all amino acids are the basic amino acid residue? Can you identify by name? Yes.
It's the K and
Anyone?
R.
K and R. Very good. K represents for which amino acid?
The lysine and R is
Lysine.
for
Okay.
arginine.
Arginine. Very good. So how many digestion it will have? Because it and none of them other are the basic, right? So M stands for. Can you please explain
Methionine.
all the amino acid? M for
The M for methionine, V for
methionine,
valine.
P for valine, D for
D I am confusing. Y is tyrosine.
okay.
G uh, this B and D are stands for either glutamine or glutamate or asparagine or aspartate. These
No.
I have known.
D is
G
for
for
oh, yes. G for
uh, G not so G T for tyrosine. And then again
yes.
M for methionine and S for serine. L for is leucine.
No, like L for leucine, yes. So D for aspartic acid. D is for aspartic acid. Y is for the tyrosine. Okay. So here now if you have the C terminal will be in the right hand side, right? So this this particular trypsin will digest at the between K and T, right? Because it will digest in the C terminal of it. So between the K and T. So that is why. And after R there is no amino acid. That is why it will not cleave at here, because it has in the C terminal of it no amino acids are there. So it will give you two fragments. One is till K and one is TML. One will be the MVDY and another one will be TMSLR. Okay, so the correct answer is 2. Now, next question is an enzyme that joins the end of two strands of nucleic acid is a polymerase or ligase or synthetase or helicase. This you might have
Ligase.
encountered in the molecular biology also while studying the replication, DNA replication and all, right?
The
So what is the
ligase.
answer? Ligase. Okay,
Ligase.
very good. So what can you please tell me the role of other enzymes also? Polymerase. What uh, what what is the role of polymerase?
The polymerase basically actually makes the base pairing, means the complementation and is complementation among the groups, that is A, T, or G, C, and helicase is used for unwinding the coil of the DNA. That is a superhelix, not superhelix.
Mm-hmm.
Helix is unwinded by
Yeah,
the
helix
helicase.
strand. Yes.
Synthetase, I don't know.
Okay. Synthetase is usually used when transfer of when joining two molecules. When you are joining two molecules with the help of energy taken from ATP, like hydrolysis of ATP, then this is called as the synthetase. Otherwise, if you are not using ATP, then it will be termed as synthase. There are the two different terminologies, synthetase and synthase. In synthetase, you will use the energy from ATP hydrolysis, and then you will join two molecule, or the assimilation of two molecules. So in that case, you will use the synthetase, or polymerase is actually the replication enzyme, DNA replication enzyme. Okay. Now, and helicase you said correctly. That is unwinding the double helix. Now, the next question is one of the enzymes involved in glycolysis, and which is aldolase, right? Now, given it, the question is given as it requires the Zn2 plus for catalysis. Okay, under the condition of zinc deficiency, when the enzyme may lack the zinc, it would be referred to as which of the state will it be holoenzyme or prosthetic group or apoenzyme or coenzyme? If it does not have
Yeah,
the
coenzyme.
Zn2 plus apoenzyme, very good, because it is given as this is used for the catalysis. That means it will act as the coenzyme or cofactor, right? So if this enzyme particularly lacks in the case of Zn deficiency, and this aldolase enzyme lacks the Zn2 plus, this state will be called as the apoenzyme. Very good. Okay, now can you please recall how many steps are there in the glycolysis?
The 10 steps.
10 step. Very good. And where does the aldolase come into place?
I think in the fourth step, that is the aldol condensation, is uh, fructose fructose 1,6 bisphosphate converted to GAP and GAP.
Very good.
It's
Very good. Yes. 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 That is the case. So now coming to the next question. The active site of chymotrypsin consists a catalytic triad with three conserved amino acid residues, and your options are histidine at 57 position, aspartic acid at 102 position, or serine as 195 position. Histidine at 107 position, aspartic acid as 152 position, and serine as 195th position. Then option C is aspartic acid as 57th position, histidine at 102 position, and serine as 195 position. Option D is serine as 57 position, histidine at 102 position, and cysteine as 195th position. So do you remember the active site of chymotrypsin? First tell me what is the catalytic triad? What does this term represent? Catalytic triad. What do you understand from this term?
The catalytic generally means three amino acids present in a active site of the enzyme in such a way that the charged groups of the that is the side chains of the amino acid take part in the acid base catalysis also.
Very good. Okay. So can you again tell me what kind of category the chymotrypsin falls in? Like which?
Histidine 57, option A, sir.
Sorry, option?
Option A,
Which?
histidine 57, FM 102, serine 195, I think so. That
Very
is the
good.
active
Very good.
site of cytotrypsin.
Yes, yes. Active site of chymotrypsin is histidine 57, while aspartic acid 102, and serine 195. Now, can you please tell me also regarding the how these three amino acids takes part into the like catalytic activity, and how they form the like which amino acid will take part into the as a nucleophile, and which amino acid will stabilize the transition state. Can you please recall that? What is the role of aspartic acid in catalytic triad? Can you recall? And what is the role of serine? Okay, no problem. So here the aspartic acid, the role of aspartic acid is that the histidine has two like proton which it can give, right? So among them, one of the proton is being taken by the aspartic acid, and because of that, in the imino group of the histidine, the nitrogen carries a positive charge. Okay, no, nitrogen carries a lone pair electron. However, that the serine here, serine occurs in the deprotonated state, in which it acts as a nucleophile. Okay, so in that catalytic triad, these are actually the chymotrypsin, trypsin. These all are the serine protease, serine proteases. So in that serine proteases, what does the serine act as a nucleophile, and then it attracts the peptide bond or any bond where the carbonyl carbon, so that the rearrangement can happen. And meanwhile, the histidine is being stabilized by the aspartic acid, because it constantly is making the hydrogen bonding with the another part of the histidine. Okay, so this will get clear when we will see the mechanism. A ma'am has also shown this, but yeah, just to discuss with you all, that is why I asked this question. Now coming to the next question. Here, your next question is the reaction is given below, in which the specific enzymes, the E is a E here, this particular enzyme E is a lactate dehydrogenase. Okay, so and the reaction is given here. So you need to identify which kind of reaction is this, which which fashion it is. Like the lactate dehydrogenase with its cofactor NADH, and it first form a NADH complex. Okay, enzyme NADH complex, and then the pyruvate comes in. So if you recall, this is the glycolysis step, right? Last step of glycolysis, when the lactate is being formed, right? Lactate is being released. So if you recall it, so here the lactate dehydrogenase takes the NADH as its cofactor, and then the pyruvate comes in. The final NADH and pyruvate complex is being formed, and then at the end you will get a product of lactate. So you need to identify which reaction fashion is it. Is it ping pong reaction, or random sequential reaction, or ordered sequential reaction, or linear reaction? What is take on this question? Any guess?
So option C.
Option C. Why do you think so?
So because
Why ordered
in this,
sequential?
so because the reaction is it cannot be ping pong, because in ping pong reaction one substrate gets react, then releases, then the second substrate gets entry. And in
Very
random
good.
sequential there is no order of reaction. Means
Mhm.
any substrate can bind at any time, either A or B, anyone. And in
Very good.
linear reactions are generally simple reactions. That's why I said
Yes.
option C.
Very good. Very good. So, because the both the sequence is defined, the first NADH should bind to enzyme, then only the pyruvate will come. That is why this is a kind of ordered fashion, and the sequence is also given. So that is why this is a ordered sequential reaction. And your explanation for ping pong and random sequential, that was also correct. Ping pong is it's you can understand that by a seesaw kind of thing. So you will have a kind of enzyme is there. Okay, then the first substrate will come, E plus S. It will form a ES complex, and then when the ES let us say this as a is the two substrate as A and B. Okay, so let us say, so it will form a EA complex. Then apart when the molecular the bonding rearrangement will happen, then the enzyme will transform to its kind of a form where it is not in the native state, and your first product will release out of it. And a part of the a part of this substrate will get attached to the enzyme. That is why it will be in the E star state, which is not the native form of E. Then the first product will release, which is P. Then the second substrate will comes in. Second substrate will come and it will form a E star B complex. E star B complex. And then this B will take the remaining part of A, which may be the transferase or any other kind of system, and then it will release the second product, and it will release the second product, which is Q, and enzymes falls back into its the native state. Okay, and thereby and it is now ready for the combination with the another substrate, binding with another substrate. This is ping pong because you understand this by the a kind of swing machine, or not swing machine, the kind of jhula. Okay, so one person will sit, then it will go to the back side. Now the when the swing is on the left hand, like on the upstream, then it is E star. Then again one person or one kind of what you say, the energy will come, and then the it will go into the another end. Means this is the main state. This is the equilibrium state. It will go to this side, which is E star. Then it will go to the another side, and then in the final state it will come to the resting state where the E. So here it is E star and here it is E star B. So similarly, this is a kind of seesaw. So that is called as a ping pong reaction. And also two substrates are required, and these are also called the ping pong bye-bye equation. Ping pong bye-bye kind of reaction. Now your next question is, in case of vitamin B6 disorders of let me clear this screen. Okay, so your next question is, in case of so the correct answer for the question was correct answer for the question was order sequential execution. The second next question is in case of vitamin B6 disorders of amino acid transamination takes place, which classes of the enzyme of this reaction? So which class of this enzyme is? So is it oxidoreductase, hydrolase, ligase, or transferase? Transamination, amino acid transamination. This is a hint.
The option D.
Option D. Okay. So option D is very correct. Very much correct. This is a transferase because the name only suggests transamination means from one molecule to another molecule, amide group will be transferred. So this is a transferase. Now the next question is NAG and NAM of peptidoglycan cell wall is cleaved by which of the enzyme? Lysozyme, polymerase, ligase, or synthetase?
Option A, lysozyme.
Option A, lysozyme. Very good. Because lysozyme is also called as the like what do you say, the peptidoglycan cleaving site or bactericidal enzyme. Bactericidal enzyme because these peptidoglycan layers are found in the outer membrane of the bacteria. So here, so when the lysozyme is usually used to cleave the NAC and NM peptidoglycan bonds. Okay. Now the next question is in the below mentioned enzyme belongs to which category? Is it oxidoreductase, transferase, isomerase, or ligase? Here the maleate is converted into the fumarate. So which category of this enzyme belongs to?
The C, isomerase.
Isomerase. Very good. Because the form is not getting changed, but the it's like the any additional group is not being attached or nothing is being changed. Only that its isomerism is being formed. So that is why this enzyme is isomerase. Very good. Next question is helicase is an example of esterase, peptidase, acid anhydride hydrolase, or phosphodiesterase. Question number 13. Which category helicase belongs to out of these four?
The option C.
Very good. Option C is acid anhydride hydrolase because it unwinds the DNA and there they take the role of anhydride and they cleave the like they just cleave the which the hydrogen bonding, right? Because the helical structure is being joined by the hydrogen bonding only. So the helicase take participate and these are the classes of acid anhydride hydrolases. Now the next question is the cofactor of pyruvate kinase is it potassium ion, nickel ion, copper 2 plus or Fe2 plus? Pyruvate kinase. This was in the lecture number 26, I guess. Okay, do you remember this? The correct answer is potassium. Next question is trypsin is an example of which class? Serine protease, threonine protease, cysteine protease, or aspartic protease? I had answered this question in previous questions. Can you guess it? Trypsin, chymotrypsin, these all. Which kind of protease? Normally, trypsin, the catalytic triad of trypsin, usually trypsin and chymotrypsin, they have the serine in their catalytic triad, right? Because of the presence of serine, these are termed as the serine protease. And I have just discussed in the chymotrypsin that it has the serine inhibits catalytic tide, which act as a catalytic, act as a nucleophile, right? Now the next question is arazinase contains the cofactor. Which one? Mg2 plus, Zn2 plus, Mn2 plus, or copper 2 plus? The correct answer here is manganese, Mn2 plus. Okay. So by this we end the problem solving session, the problem practice on module 6. Now we will be discussing about the scope of this module with the research perspective and which all techniques you can use to enhance your research skill. So before that, I would like to ask if there are any other doubts or any concern related to the module till now, whatever have been covered in this course. If there are any doubts or any question related to it, you can unmute yourself and ask. Okay. Try to interact. Are there any question? Ranjit, Swarnama, or Jayshree? Any question or any suggestion that you would like to give
No,
to
sir.
if okay. Jayshree, Ranjit, okay. You can use the feedback form also to give your comments and if there are any suggestions related to it. Let me discuss with you all related to the if how you will because till now you have been in the module 5 you have been you have been seeing that the catalytic triad or any kind of active site is very much important to predicting the function of a protein or to analyze the catalytic activity like the its catalytic mechanism. So to identify the catalytic mechanism, you must understand which kind of active site amino acid residues are present in this in particular kind of protein, right? So, I would like to tell you the technique how you can identify the active site using the computational techniques. So here in the computational techniques, you don't need many softwares. You just need few of the softwares or and the things that you will need the structure file pdb structure file protein data bank file of your particular protein which you want to study. Then if you want to study uh, according to particular ligand in which uh, what is the binding site of this ligand in that particular protein, then the first step is to procure the pdb file of that particular protein, then the mol2 file or any format in htf format, and then you convert them using the open babel that I discussed in the last discussion class. So you just get the structure file of these two, then you perform the docking using autodock tool. I have already given the protocol related to the autodock in the last email that I had shared. So there I had shared the complete protocol of the performing the docking simulation. Then what you do, you perform the docking and then the final result file of the docking is .dlg file, right? You analyze that dlg file and identify which is the energy minimized structure, the less energy structure of that, and then you get the pdb file of that. You convert that once you get the dlg file out of the autodock, then you will open that in the analyze tab of the docking and then in Autodock, and then you go to the lowest energy structure or lowest energy conformation of the boundary complex, that ligand and protein, and then you convert that file into the pdb file. Now what you can do, you can extract that pdb file and then you can analyze into the PLIP. PLIP is the protein ligand interaction profiler. This is a web tool. or else you just want to know which all are the probable docking site of a particular protein, then you can directly submit that into the P2 rank, that is a web server. So I will show you how you can perform this task in my uh, like browser. So the first thing you want to check, you want to download the protein structure file, right? That you can download from the RCSB PDB data bank. There you can, if you know the protein PDB ID, then you can type the PDB ID directly, which is this 4-letter code, 4CHA or any 2CHA. There are different type of the different structure, crystal structure of a particular protein. Every protein have been assigned a 4-letter code which is called as the PDB ID. You can search using the PDB ID or you can search by name. Okay, so let me now explore what all the things you will see in a PDB data bank. So if you open any kind of structure, let us say we are opening the 4CHA. 4CHA is for the chymotrypsin protein. It is opening, and then I will show you which all the meta information you can get from the PDB ID. Let it open. Yeah, so it is open. Now the first thing that you can understand here, the structure of alpha chymotrypsin refined at 1.68 angstrom resolution. So what does this mean? Like, so 1.68 angstrom resolution. This is nothing but the lowest when you are studying you are studying the structure of a protein using X-ray diffraction method. These resolution are given in the angstrom values, because if you remember the concept of XRD, these are normally given as the layered structure, right? So in that, if the the lower the resolution, the more accurate the structure will be. So if you have two or more structures for a particular protein, then you identify the lowest resolution of that structure. And this is not only the uh, deciding criteria for a consistency of a protein. The another things are the class score, Ramachandran outlier. Here the Ramachandran outlier are in the very poor range. You can see here the blue represents the better and the red represents the worse, right? So the more you find the parameters in this blue region, the more accurate that structure is and more reliable that structure is. You can download the structure using you going to this tab. You can download the faster sequence of this protein, or you can directly download the PDB format of this structure. So this PDB format, this particular PDB format is important. This is particularly required for any kind of study that you want to carry forward. Okay, the other information you can get here, right, in the metadata information. So here it is given as this particular this particular protein has a 50.53 kilodalton. This is the molecular mass. This has the molecular total weight of this particular protein is 50.3 kilodalton, and atom counts are 3,500, around 3,600, right? The amino acid residues counts are 482. Deposited amino acid residues are 482, and modeled amino acid residues are 477, right? And all other information that you can get, the hydropathy index, disorder binding, then what all things are bound into this are the protein B chain has the 133 amino acid length. Then this is the mutation study, and the EC number is given here, which is 3.4.2.20.1 Then the amino acid alpha chymotrypsin A chain has the 97 sequence length. So these all information that you can get from the PDB data bank, right? Now I wanted to tell you the if how you will check the binding pockets of this amino acid. What you can do, you can go to this P2 rank or PrankWeb website. PrankWeb website is for the ligand binding site prediction without docking. Okay, if you don't want to do docking, then you can directly go to the PrankWeb website. You deposit. You you can directly write the PD. You have three options here. If the structure is already available in the PDB data bank, you can directly write the PDB code. Or if you have designed the structure by yourself, then you can upload the structure file, PDB file of this, PDB file of your particular what you say the protein. And then you can, if you want to restrict it to the particular chain, if you have many chains, A, B, C, D chains are there in your protein, then you can restrict it to the chain number also. Or if you have the predicted structure, then you can get the uniprot ID also. Okay, so I am predicting the binding pocket of this particular chymotrypsin itself. So I will just copy paste the 4-letter code of this PDB ID of this particular amino acid, this particular protein, and then I will paste it the PDB code of this. Then you just hit submit button. Then this web server will give you how many pockets are there in the like here. So there are total of how many pockets? 7 pockets are there, and all the pockets have been given a particular kind of score and probability score also. The more the probability score is there, the more reliability of that particular pocket will be. Okay, so there are, and if you want to visualize the, see here the different color codeds are given as the different pockets. You can also check. So here the first pocket is this blue one, right? Then the orange one is here, right? Then pink one. There are different pockets. If you want to visualize it in the different fashion, so this is the that particular chymotrypsin amino acid, chymotrypsin protein, and these color coded are the binding probable predicted binding pockets of the of this of this particular protein. If you want to visualize only this pocket, then you just click on this other all will be disabled, and you will be able to see only blue pocket. You zoom in, you can zoom in, or for better visualization, what you can do, you can show the protein visualization as ball and stick, and then this as the surface. Okay, then you will be able to see here it is the binding pocket of this amino acid, and these are the amino acids that you can see. Oh, okay, so these are the amino acids here listed here, the pockets, and from here you can identify which all the amino acids are there. Okay, if you want to check like check which all amino acids are involved, then you need to download this file in any format. There are 3 or 4 format, like zip archive is downloaded. Then you download this and open this folder. Now you have CSV file and visualization both. Okay, if you open this CSV file, this will give you the all. Yeah. Let me open this. This will open in the Excel format, and then there are all 7 pockets denoted here. Okay, so in that pockets, there are in the B chain. The first pocket is in the B chain. Okay, B. All amino acids are in the B chain. C chain also there. Then F chain. There are multiple chains in this particular protein, right? So for that, what you need to do, you copy paste, copy these all coordinates. These are nothing but the 141 amino acid residue. So you check in the PDB file of that 141 position which amino acid is there, and then you can identify this particular amino acid is the binding pocket of the this particular amino acid is involved in the pocket one. Okay. Likewise, you can predict the binding pocket of any given amino acid of any given protein. Okay, understood? The another technique is what I have told you here. You just run the docking simulation and then perform the docking, analyze the DLG file, and then upload that into the PLIB. Let me show you how the PLIB result will work. If there is anything open here, no, I guess. So I will show you my file here. PLIB is this site, protein ligand interaction profiler. So here you either you can give the PDB ID. Let me give you a PDB ID of 1 TCA. Okay, and then hit enter analyze. Then this will open. Okay, this will open. Now it is showing which all amino acids are forming which kind of bond. So here in the hydrogen bond, this but 1 TCA is for NAG compound. Here NAG compound is attached into the 1 TCA compound, which is lipase. 1 TCA is for the lipase, and this is from the Candida antarctica lipase B. Okay, it has two ligands, which is NAG, which is NAG B1 and NAG B2. Two NAG are there. You can see here, and then these are the amino acids. You can also visualize this into the 3D model. This is loading, and here you have two types of bond: hydrogen bonds and water bridges. So here you can see which all amino acids are involved in bonding. ASN represents for the asparagine, ASP for aspartic acid, then GLN glutamine, say SER serine, right? So these all, and this is the NAG group. This is the ligand, and ligand there are different bonding. So this is water bridge. Blue one is the hydrogen bonds, hydrogen bonding with the oxygen and nitrogen, right? So here you can also see the glutamine at the 11th position is 2.3 2.32 angstrom distant from that, and it is the HA means hydrogen acceptor, and distance of the hydrogen donor is 3.22 angstrom. The donor angle is also given. The protein is donor and side chain is also donor, right? So and the donor atom is NAM, right? So these all information you will get. So for hydrogen bond formation, you will have GLN amino acid. GLN stands for, can somebody tell me? GNL is short form for?
The glutamine.
Glutamine, very good. So for hydrogen bond formation in lipase amino acid residue, a lipase protein, the glutamine is forming the hydrogen bond with NAG ligand. Then asparagine is there. Then aspartic acid at 75th position and aspartic acid at 75th position. For in the water bridge, serine is playing a role. Serine 10. Then aspartic acid at 75th position and glutamine at 77th position. Okay, understood? So using these two tools, you can identify the binding pocket and type of bond that are being formed. Okay, so this all things I would like, I wanted to tell you regarding this module. So I hope this was useful for you, and thank you so much for joining the session. If you have any comments or any suggestion, you can write down that in the feedback section, or else if you have any question, you can ask, you can email that to me, or you can also directly post your question in the forum. Okay, so by this, the session for this week 6 is now reaching to the end. So thank you for joining. Have a a very good day and happy learning. Thank you so much.
Thank you, sir.
Any question?
Sir, I have a question.
Yes, please go ahead.
For the software now, before
Yes.
running Discovery Studio, we have to run with some program. Then
Mm-hmm.
only we can visible those protein binding sites.
Mm-hmm.
This is a autodoc now. So before that, we have to run some program. I have learned that, but I
Mm-hmm.
forgot what we have to run. Before
Mm-hmm.
visualizing in Discovery Studio, we have to run that program. Then only the protein where our ligand bind with our protein is the exact
Yes,
active
yes,
site will be appear. So will
yes.
you
That
share me that program code?
yes. I had shared that in the previous session actually. Let me share my screen once more. Let me share my screen. Then I will tell you.
Yes, sir.
Actually, I had shared the full document with you all that you can see in the forum. Like every Friday, I used to give you the links, right? So here the docking protocol. If you click in this link, you will get entire docking protocol. How you perform the docking. You have the entire protocol for this, like ligand structure minimization, then the docking preparation, or performing the auto dock. Everything you will get here. Okay. Or
Okay,
else, or else if you want the complete document, then I have also given that in the resource folder. So in that resource help manual, you will get how to perform the docking, and then docking you can go through this docking one. Okay, this tutorial. This is 3 hour tutorial, and then this is really good tutorial. He like the resource person will tell you each and every step in very explanatory way. Or other software also, if you want to learn, that also have given the link here. Okay, all the
okay,
Python learning and Kuta method, then different methods are there. Okay,
okay. So let me
so
go through that.
yes, you just go through the step by step procedure of this. You will be able to for you will able to follow the complete docking protocol.
Okay, sir.
Okay.
Let me
Oh,
once again I go through with those softwares.
sure. Okay. Any other doubt?
No, sir. I'm
Okay.
good.
Thank you, Jesse.
Thank you, sir.